Hi guys, this is the cell. All right. Um, so cells come in many different shapes and sizes. Um, this kind of right here is kind of showing kind of like maybe what you'd see on some high-powered microscope. You can kind of see in the red. Those would be maybe some nuclei. You might be able to see some cytoskeleton, some stuff like that in there. This maybe is a little bit larger of a cell, maybe some sort of paramecium or amoeba type critter. Pretty small, gonna have a nucleus, but actually a pretty decent cell size. Okay, you can see some of these spots. Those are going to be maybe some typical like organelles. All right, that the cell might have that kind of have certain jobs, and we'll talk more about that. And then right up here we got a neuron. Okay, they are quite long and skinny. They are, they're found in your brain. They transmit nerve impulses. The longest cell in your body, it goes kind of from the tip of your tailbone all the way down to your big toe. They can be, not as one single cell, so they can get pretty long. Also, if you look at these light blue boxes, those are another different type of specialized cell called Schwann cells. All right, that kind of help increase the speed that this nerve impulse travels. So they come in all sorts of shapes and sizes. I think that the cell is one of the toughest things to kind of maybe put your finger on without just talking about what a cell is. We all kind of know what a cell is, but describing it can be difficult. Excuse me. Cells are the basic units of life, right? It's kind of like the atom for chemistry. This is just the basic unit in which the cell does things, where the body does things, right? It's the basic unit. Okay, they can have various different jobs, shapes, functions. Okay, a, a single bacteria cell that is one organism, right? Very small. That is one organism. Okay, you're made up of about 100 trillion cells, which is pretty crazy. Okay, and each of them are going to have a lot or many of these features. Okay, and like I said, all sorts of shapes and sizes. The cell theory. This is a theory in biology, put together by a few scientists. There's three tenets to the cell theory. All living things are composed of cells. So, for example, bacteria are not considered living because they are not made of cells. That's one of the strikes against them. Now, there's a couple other strikes, but that's one of the big ones. Cells are the basics, units, and structure of function of living things. We start from a single-celled fertilized egg. Sperm meets egg, two separate cells that kind of combine to form what we call zygote, and then that grows and divides and becomes you. So, and all things are made of cells, all right, to at least some function. So, for example, if you look at wood, all right, a lot of the layers inside, like that, all that wood isn't necessarily living cells, right? There's some parts of a living tree are wood, are living cells, but a lot of it's dead cells that are kind of left behind. The skin that kind of covers your body they're dead cells, but they still are cells. And then new cells are produced from existing cells. Now that begs the question, where does the first cells come from? Another story for another day, but that's all cells come from existing cells, and that kind of disproves that theory of spontaneous generation that we were kind of talking about in kind of the intro to bio section. So, how do we study cells? First off, we're going to use a light microscope. Okay, this works by passing visible light through a specimen, such as microorganism like a bacteria, or a thin. That's not a think slice of animal or plant tissue. Okay. So, if you remember, if you took A and P or taken A and P, right? We'd spend a lot of time looking at tissues, and we looked at onion cells, right? But the idea is you have to have a pretty thin slice of tissue, right? Because light has to be able to pass through that cell, right? If it's too thick, light won't get through and you won't be able to see anything. It's just going to look like dark, right? So the idea is you kind of have to cut it thin. So actually, a lot of the times, like scientists or the people that put together those tissues, what they'll do is they can cut, all right, cells, at different angles to kind of look at the cell in a different way. So this right here is kind of a neuron, right? You can kind of cut it like this, and you can cut it like this, hamburger style, hot dog style, whatever, right? And you're going to see different shapes. Is it the same cell? Yes, it's the same cell. It's just you're looking at kind of it from a different lens. It's kind of like you're using a whole bunch of 2D images to put together a 3D picture of what that cell is. Because you kind of almost have to look at things kind of in the 2D, all right? You are limited by the wavelength of light, which is about 0.2 micrometers, okay? And then right up here is kind of how microscopes work, and we kind of, you might see me messing around with some of the settings and stuff like that, but basically this is how they work, right? You have your light source, right? It can be a mirror, it can be a bulb, and then it gets into a condenser lens. That lens basically kind of focuses the light into the sample, right? And you can kind of adjust how much light actually hits the sample too. Okay, then it kind of goes into the objective right here, kind of the bottom lens where it's the light's kind of dispersed, and then it kind of is focused again, and then it goes to your projector lens, and then it kind of goes out through the eyepiece, and then you see it through here. Okay, it's not an optics class, but this is basically how they work. So just kind of interesting. Just kind of know what's on the slides. You don't have to memorize this or label this on the test. So kind of cool stuff though. So how do you get below that 0.2 micrometers or the 2 nanometer level? So you have to use electron microscopes. So we can what we're doing is we're basically shooting electrons at things, all right, to observe specimens. And I'll throw something on the board on Monday for it when I get back. So you can get down to about 2 nanometers, much smaller than those light microscopes. So you kind of have 2 types of electron microscopes, big ones. You have your scanning electron microscope. This studies the detailed architecture of cell surfaces. So for example, these aren't cells, or not exactly. This is pollen granules, so they're kind of the outside of, like plant sperm. Okay. So this right here is, it tells you a very good idea of what's on the outside of the cells. It does not tell you what's really on the inside, but it tells you, what's surrounding. What does the cell, what does the structure look like, if you're looking at it. Okay. This, it's kind of like almost like an HD. It's really cool to see some things. You can see like, scanning electron microscopes of like bugs and stuff like that. It's really cool. A transmission electron microscope studies the details of internal cell structure. So this right here is a transmission electron microscope. Okay, so you can kind of see like the organelles, right? This for sure is nucleus. Your nucleolus right in here, right? Some you see some of the cell membrane and wall and outer parts of the cell. So these are both tools that scientists use. They would just use them for, different purposes, Sometimes you use a hammer, sometimes you use a saw. It just depends on what you're doing, Cell sizes. Cells are going to range from 5 to 50 micrometers in diameter. The tiny cells are bacteria that are only about 0.2 micrometers. And then the largest cells are the giant amoeba, which is 1,000 micrometers, or if you look at a millimeter, that would be about the same as a millimeter. So if you look at a ruler, the little tick, the tiniest little tick, right, that's about a millimeter. So they can actually get pretty big across. We looked at them in college, and you could actually kind of see amoebas and critters like that move actually across the little slides. They don't look like much. They're pretty clear. Unless you were to dye them, but if you wanted to keep them alive, so you can actually see them crawl around, and they're pretty cool to see. So, and we will look at bacteria this year. We'll stain them and look at it under a microscope. Maybe we'll throw some amoebas if I can get around to it. We'll see. There are 2 types of cells. There are 2 broad categories of cells. You get your prokaryotes and your eukaryotes. How I remember the difference is you are a eukaryote. Okay, prokaryotes are stuff like bacteria and archaea. We'll talk about that. So prokaryotes, they lack a nucleus, they have no membrane-bound organelles, and they are generally smaller and simpler than eukaryotic cells, generally speaking. Okay, the examples are bacteria, archaea, which are these extremophiles, things that live in places like the hot springs at Yellowstone, as well as some algae like cyanobacteria is a type of bacteria. But really small stuff that you can't see are prokaryotes. Everything that you probably think of as life, all right, like trees and plants and mushrooms and all the animals, right, as well as some other things you can't see, those are eukaryotes, okay? But archaea, they're cool. We'll talk more about them, but they're kind of the extreme guys. And then just overall, they are smaller and simpler. The no membrane organelles, everything's kind of a soup in them. We talk about them in when we go over a unit in microbiology, but everything just kind of mushes together, okay? And there is a point of organelles, and we'll talk about that. And they don't have a nucleus, and that's cool too. So eukaryotes, flip side, they contain a nucleus. They're generally larger and more complex. They have the membrane-bound organelles. This is plants, animals, fungi, some algae, some other microscopic organisms like amoebas, like parameciums, right? If you look at, I believe some of the posters on my wall, I have a lot of those guys are going to be eukaryotes, actually, that pond series, okay? A lot of those guys are going to be eukaryotes. Even though they're stuff you can't see, they're usually a little bit bigger than bacteria, quite a bit bigger. Not always, but those are going to be your eukaryotes. Remember, you are a eukaryote, okay? You have a nucleus, okay? That's just how I remember. So let's talk a little bit about the function and the purpose of having organelles. Okay, what's the point of having a mitochondria? A bacteria doesn't have it. Why doesn't it need it? Okay, we're a little bit more bigger and complex. The point kind of organelles is that what it does is it allows for things to be kept separate, especially chemical reactions. Okay. So for example, we looked at hydrogen peroxide. We did a liver lab right when you guys were sophomores in biology. But this right here is hydrogen peroxide. It's a pretty reactive thing. Okay, we can kind of keep this thing separated, okay, in a separate organelle called a peroxisome, okay, where it is not going to affect other parts of the cell and chemically damage it, okay, and it's kind of contained in these phospholipid bilayers, okay. This right here is a mitochondria, okay? It's going to have a couple of layers, all right? It's going to have a layer here, right? It's going to have an internal membrane here. It's going to have another membrane on the very outside of the entire cell, and there's different chemical reactions that happen in different parts of the mitochondria and of the cell, right? This right here is looking at cellular respiration, okay? About 3 big steps. You have glycolysis, the Krebs or the citric acid cycle, and the electron transport chain, okay? These 3 things are going to happen in different locations, and they are going to kind of use these, the walls, the barriers of the organelle to kind of keep these reactions separate, okay? So for example, we'll talk how it's really important for the electron transport chain to kind of use this barrier, and this is actually how it pumps, hydrogen ions through and stuff like that. But it kind of keeps things separated, okay, with these plasma or phospholipid bilayer membranes, right? Also, if you have an enzyme for glycolysis and you're doing it in the cytoplasm or the cytosol, right, there's no need for that enzyme to go in the mitochondria, right? There's no need for it to go, in the very inside of the mitochondria, right? Because glycolysis is happening here, okay? So you only need to make it, and you only need to put that tool in this area. Same thing with the Krebs cycle, same thing with the ETC, okay? We'll talk more about this process as the year goes on, but organelles keep reactions separate. Okay. A lot of, biologists still kind of look at the cell as just a big collection of chemical reactions, okay, caused by enzymes. Okay. So these organelles kind of keep these reactions separate. Okay. I am yapping a lot. Y'all have a fabulous weekend. We'll call it a day. How do I stop this thing? That's the key.